NAD+ precursors such as NMN, nicotinamide mononucleotide, and NR, nicotinamide riboside, have emerged as some of the most promising compounds in the field of healthy aging and cellular rejuvenation. Though they are not direct telomerase activators, their role in supporting telomere integrity is significant, particularly through the enhancement of mitochondrial health, reduction of oxidative stress, and activation of key longevity-related proteins like sirtuins and PARPs, poly-ADP-ribose polymerases. As NAD+ levels decline with age, restoring this critical coenzyme through precursor supplementation may offer a foundational strategy to maintain telomere stability and slow biological aging at the cellular level. NAD+, nicotinamide adenine dinucleotide, is essential for a wide range of cellular processes, including energy metabolism, redox balance, and DNA repair. Among its most important roles in the context of telomere biology is its activation of sirtuins, specifically SIRT1 and SIRT6. These enzymes regulate chromatin structure, suppress inflammation, and stabilize telomeric DNA. SIRT6, in particular, is directly involved in maintaining telomere structure by facilitating proper DNA packaging and preventing degradation of the telomeric ends. In mice, loss of SIRT6 leads to telomere dysfunction and premature aging, whereas overexpression preserves telomere length and extends lifespan. By replenishing NAD+ stores, NMN and NR supplementation can restore the activity of these sirtuins and thereby improve DNA repair and telomere protection. In addition to sirtuins, NAD+ is also required for PARPs, enzymes that sense DNA damage and initiate repair. Telomeres are especially vulnerable to DNA strand breaks and oxidative lesions, and impaired PRP activity has been linked to increased genomic instability and accelerated telomere shortening. Thus, maintaining optimal NAD+ levels supports a robust repair system that guards against telomeric attrition. One of the key mechanisms through which NAD+ supports telomeres is indirect but critically important: mitochondrial rejuvenation. Dysfunctional mitochondria generate excess reactive oxygen species, ROS, which damage telomeric DNA, already a sensitive region lacking the robust repair mechanisms available to other parts of the genome. By improving mitochondrial efficiency and biogenesis, NAD+ precursors reduce oxidative stress at its source. Healthier mitochondria also mean better energy availability for DNA maintenance and cellular renewal, further promoting telomere preservation over time. Preclinical studies in rodents have shown that NMN and NR can extend lifespan and improve numerous markers of biological aging. Treated animals exhibit enhanced physical endurance, better cognitive function, and improved metabolic health. Importantly, they also show reduced signs of cellular senescence and DNA damage, conditions intimately linked to telomere shortening. While direct measurements of telomere length in these studies are still emerging, the broad systemic improvements suggest a more favorable cellular environment for telomere maintenance. In humans, trials are now underway to evaluate the long-term effects of NAD+ precursors on aging biomarkers, including telomere length. Early results are encouraging. Supplementation with NR, for instance, has been shown to raise NAD+ levels in blood and muscle tissue without adverse effects. Small studies suggest potential benefits on cardiovascular health, inflammation reduction, and insulin sensitivity, all of which are factors that influence telomere erosion indirectly. As these studies scale and include longer follow-ups, it is expected that more definitive evidence on telomere dynamics will emerge. Moreover, NAD+ precursors are synergistic with other lifestyle interventions that support telomere health. Intermittent fasting, exercise, and polyphenol intake, e.g., resveratrol, all increase NAD+ availability or activate sirtuins. Combining NMN or NR supplementation with these practices could amplify protective effects, not only supporting telomere length, but enhancing systemic resilience to age-related decline. An additional, often overlooked advantage of NAD+ precursors is their potential to enhance stem cell function. Stem cells are highly sensitive to metabolic cues and DNA integrity, and their regenerative capacity diminishes with age in part due to telomere shortening. Boosting NAD+ has been shown to revitalize aged stem cells, especially in muscle, neural, and hematopoietic systems, potentially preserving their ability to repair and regenerate tissue. This could reduce the pressure on somatic cells to proliferate and thereby help conserve telomere length across multiple cell populations. In conclusion, while NMN and NR do not directly elongate telomeres, their ability to boost NAD+ levels touches nearly every process that safeguards telomeric integrity, from DNA repair and mitochondrial health to inflammation control and sirtuin activation. Their low toxicity and growing clinical support make them one of the most accessible and biologically plausible tools for protecting cellular health as we age. As science continues to unravel the intricate connections between energy metabolism, genome maintenance, and telomere biology, NAD+ precursors may become a cornerstone of future longevity protocols, strengthening the internal scaffolding that keeps our cells youthful and resilient for years to come.